Good day everyone, we are from Group 24 and today we're going to talk about the draft genome of the specialist flea beetle Altica viridicyanea, Coleoptera Chrysomelidae. Host-specific herbivores insects often diversify quickly due to shifts and specialization onto distinct host plants which promote reproductive isolation and speciation. The genus Altica exemplifies the process. Three closely related species, Altica viridicyanea, Altica crucicola, and Altica fragarii, are morphologically similar and sympatric but feed on distantly related host plants from different families. This dietary shift is a key driver of their rapid divergence. Adaptation to new host plants in Altica involve full behavioral changes to recognize and select new plants and physiological adaptations to detoxify different plant secondary compounds. Gene families related to chemosensation and xenobiotic detoxification are critical in these adaptations. Among these species, Altica viridicyanea is an extreme specialist feeding exclusively on germinal balance. Its reproductive isolation is mediated by species-specific cuticular hydrocarbon that influence mating preferences. Interestingly, these chemical profiles are also affected by diet linking host plant use directly to reproductive behavior. This study presents the first genome assembly of Altica viridicyanea, making the first for the Alticinae subfamily and the only fifth of the Chrysomelidae family. Unlike previously sequenced chrysomelids, which are oliphagous, Altica viridicyanea is monophagous, making this genome a valuable resource for understanding the genetic basis of host plant adaptation and speciation. The first objective of the study is to generate a high-quality draft genome assembly of Altica viridicyanea. The primary objective was to produce the first genome assembly for the subfamily Alticinae, providing a robust genomic resource for the specialist flea beetle which feeds exclusively on a single host plant species. The second objective is to annotate the genome comprehensively, including repetitive elements and gene families, particularly those involved in chemosensation, detoxification, and plant cell wall degradation, which are key to host plant specialization. The third one is to provide a genomic resource to facilitate comparative and evolutionary studies. This will help uncover molecular evolutionary processes involved in ecological speciation, particularly how shifts in host plant use drive genetic divergence and reproductive isolation. The last one is to support future research on the genetic mechanism linking host plant use and speciation. The genome resource is intended to facilitate detailed investigation into the genetics and molecular mechanism underlying behavioral and physiological adaptations to specific host plants.
Next, I will present the methodology for this experiment. This experiment was started by creating a laboratory colony. This is done by collecting adult viridicyanea and maintained in grow chambers at 25 degrees Celsius and fed with leaves from the host plant, Geranium nipalensis. Before sequencing the whole genome of these beetles, the researchers estimate its genome size using flow cytometry. We prepared a cell suspension in gallbladder buffer by dissecting thorax muscle from 4 males and female live beetle. The tissue is ground, filtered through a 40 micromillimeter nylon mesh, and collected in chilled tubes. Then, the nuclei was stained with propydium iodide and incubate the samples in the dark at 4 degrees Celsius for 2 hours. Fluorescent intensity is measured using a MoFlow XDP flow cytometer, and genome size is estimated by comparison to chicken blood as an internal reference. Next, the researcher extracts genomic DNA from whole beetles using the DNeasy blood and tissue kit, following the manufacturer's instructions. DNA quantity is measured with a qubit fluorometer and quality is checked using agar gel electrophoresis with high molecular weight ladders. To support gene prediction, the researchers generate transcriptome data using PacBio and Illumina platforms. The researcher used whole bodies of 4 males and 4 females for RNA extraction and create 3 PacBio libraries yielding 8 GB of clean data. Additionally, the researchers generate 10 GB of clean Illumina RNA sequence data. Due to the small size of the beetles, whole adults are used for sequencing. The researcher assessed the quality of Illumina raw reads with fast QC and trim adapters and low quality reads using trim galore. The researchers estimate heterozygosity using GCE based on pooled Illumina reads from submitted females. Next, the researcher assembled the genome using a hybrid strategy. First, PacBio reads are assembled with fly to generate contigs. These contigs are scaffolded with Illumina reads using redundants, which utilize SS space. SS space is then used to further scaffold using PacBio long reads. The researcher then evaluate assembly quality by mapping PacBio RNA sequence reads to the genome using Minimap. Poorly mapped transcripts with coverage below 0.99 and identity below 0.95 are further analyzed with the CoGen pipeline to assess potential function. The researchers evaluate genome completeness using BUSCO against Insecta and the Endopterygota datasets. To further assess gene completeness, the researchers compared protein lengths with those of tree beetles with high-quality genomes, which are Tribolium castaneum, Dendroctonus pondens rosea, and Anophlora glabri-penis. The researcher identified repetitive elements using LTR Finder, Repeat Scout, and PyLer-DF. Repeats are classified with PEST classifier and merged using RIP-based database. Repeat masker is used to mask repetitive sequences in the genome. Gene prediction combines de novo, homology-based, and transcriptome-based methods. Non-coding RNAs are annotated using RFEM and MIR-based. Lastly, the researchers reconstruct the phylogeny of A. viridicyanea and 9 other beetle species using 1,751 single copy autolog.
Hi, my name is Nur Wahida and my metric number is A196097. So I will talk about result and discussion. Next, reading and assembly using Illumina and PrepBio, the found total size of the assembled genome is 864.8 MB with high number of scaffold and N50 is 557 KB. So what does it mean? It means that N50 is the length of the scaffold or contig at that half of the total genome. N50 is high in this case. So it is indicate this is a well-assembled genome with fewer fragments. And even though the assembly shows quite decent quality, the high number of scaffold still suggests there are gaps in this assembly. Right after our assembly, we want to check the completeness of our assembly. We use BUSCO. So the way it works is like a checklist of important genes that we expect to find in the genome. In this case, from table 1, they use two different checklists for insect checklist and Endopterygota checklist. Inside the table, we can see the percentage of complete BUSCO for both insects and Endopterygota is very high, meaning nearly all key genes expected for insects and Endopterygota are present and mostly come in single copies. About the missing BUSCO, the percentage for both is really low, around 1 to 2 percentage, meaning that it is successfully captured all expected gene and has very low missing rates. For part 2, genome annotation, first we will see about repetitive sequences that cover 62.9% of the assembly. Table 2 shows the composition where it mainly retrotransposon and then A transposon. And among retrotransposon, line, gypsy, and lard were the most abundant, while in the A transposon, TIR is the most abundant. And they need to check the repeat before genome annotation because repeat can confuse gene prediction. Later, they found the total protein coding gene 17730 and non-coding RNA model 2462. For part 3, phylogenetic analysis, we look at figure 2 and we see that they compare A. viridicyanea gene with 9 other beetle species and found so many share autolog, showing strong conservation. And then it put D. viridifera and L. disseminator close to A. viridicyanea that they say it have divergence estimated around 74.7 million years ago. And notably, 155 gene families expanded and 27 contracted from A. viridicyanea gene. And this highlight possible adaptation to environment. Under plant cell wall degradation, the key takeaway special from this section is that A. viridicyanea has fewer types of plant cell wall degrading enzyme. It have around 65 GH genes, but other beetles have more GH genes. So this is indicate the limited ability to digest diverse plant, helping explain its monofocus specialization. Under common sensation, it shows that GR that shows something unique compared to IR, OBP, and CSP gene. This is because in most beetle, when you compare the GR genes between species, it is usually a mess, meaning the gene don't match up neatly across species. Scientists call this as simple autology, is rare. But in A. viridicyanea, the GR genes are clear and well defined. They can tell apart carbon dioxide, sugars, and bitter substances really well. This kind of thing will make chemosensory system more specialized for detecting important cues from its host plant. Finally, for detoxification supergene families, usually herbivore rely on many detoxification enzymes to break down or neutralize harmful plant chemical. In this case, P450 and CCE is not really unique compared to GST and ABC transporters that show more interesting pattern. For GST, they are divided into groups like epsilon, and A. viridicyanea has only a few of these. This means it is not very efficient at detoxifying many kind of chemicals, which explain why it is monophagous. And for ABC transporters in A. viridicyanea, they seem specialized to deal with toxin from just one plant, meaning they help to remove toxin, but it only from the very specific host plant.
My name is Aina Shuhada Tirasi. From the genome annotation, we found that Altica viridicyanea is a host specialist herbivore that has a reduced number of chemosensory and detoxification genes as compared to more generalist herbivorous beetles. Although Altica viridicyanea had fewer chemosensory identification genes than more polyphagous beetles, we did observe an expansion in some gene families that may be related to host plant adaptation. The genome assembled here provides an important resource for further studies on host plant adaptation and functionally affiliated genes. Additionally, this work will also open the opportunity for cooperative genomic studies among closely related Altica species and may provide insights into the molecular evolutionary processes that occur during ecological speciation.